Good evening, good afternoon, and welcome. It's a pleasure to start the new year, and we start the new year with a special guest that started ELSC long time ago, and he started ELSC because ELSC would not have been here without ICNC. So will be our guest, but before we go to our guest, we have a small ceremony. So as you know, ELSC is the Edmond and Lily Safra Center for Brain Sciences. A year ago, or two, was it? Shimon Peres was 90 years old, right? He had his birthday. It was 2 years or 1 year? So it's 2 years ago, right? And then, if you remember, we had an exhibition in the Minyane Hauma, etc. And the money came from Safra for this exhibition. But it also came from us because Lily Safra, the initiator of the whole thing, promised us $1 million special prize. 250 went for this exhibition, and the rest went to us. And today I am happy to introduce or to let you know about a new kind of prize we have here, the Shimon Peres Doctoral Fellowship of ELSC. And every year a new student will have this special fellowship. And this year the student is Adar Adamski. So to be honest, we chose this year by grades, and we thought after I saw Adar here many nights, we thought, wow, this was a good choice. It was not my choice. It was the teaching committee headed by Elin Elken and the new committee, right? So the new committee is Elin Elken, Adi Mizrahi, and Jonathan Levinstein. And Merav. So we are 4 in the committee now. And Merav, they have chosen Adar to have this prize. And Adar, I think you deserve it. Thank you for being what you are, and good luck in your work. Now, I would also like to thank today Ishai Gdalia and Amit Vinograd, who are organizing the seminar. And Amit came to me outside and said, maybe you should introduce the speaker today, but he didn't show himself. Amit is here. And they already do great work because we have interactions more than I ever remember, and we are really taking care to make this seminar as good as it can be. And as you can see, it starts. Eli likes to say it can never get better, but I like to say it will be even better. I don't know if it's possible. So today we have Moshe Abeles, who, איך אומרים גילוי נאות? In English, I don't know. Full disclosure? Is my PhD mentor, advisor for many years, because my PhD was many, many years. And I think even during my PhD, I remember talks or discussions about the future of ELSC and not the future of ELSC, about the future of computational neuroscience. About the future of computational neuroscience, I was overhearing discussions of Moshe with the rector at the time, who was Gidi Shavsky, I think, right? So it's long time ago. And Moshe took the mission very seriously and successfully since 1992, I believe, till retirement. And then he moved to Bar-Ilan to establish one more brain center in Bar-Ilan, the Gonda Center. So I'm really happy to have Moshe here, and I'm really surprised to have Moshe here talking about what he's talking about due to our history. And I'm looking eagerly to hear what you have to tell us about sequence of cortico-cortical interactions specific to cognitive processes. This is going way higher than we did in 1975 or so. Thank you, Moshe, for coming, and we are eager to hear you.
Thank you, Alan, for this kind introduction, and thanks to the organizers. I'm really thrilled and a little bit shaky to be here again, because it's now 20, I don't know how many years, since a group of friends with Chaim Sompolinsky and late Danny Amit and Hanoch Gutfreund, we thought something like this for computational neuroscience should be made. And nobody wanted to take over the managing the thing. And I said, I'll take it, because in 3 years, there will be a building where everybody would sit. So congratulations that there will be a building. Will be a building, okay, but good. And now for the talk. For many years, as you know, I've been doing I was doing multi-single unit recording in animals, and so first I want to thank my 2 PhD students in Bar and Idan. I'm using a lot of their data in what I'm going to show, and also my own data, and Yuval for help with the recording, and Ohad with the presentation. Now, we are all used to see pictures like this one, where oxygenation of blood is somewhat higher. You can see it's something like 1% higher during one task relative to another task, and everybody says we know that not everything in this task is done here, but here is something special about this task. Now, the problem is in this kind of thing that it gives you the image, although everybody knows it's wrong, that there is a place for every function. It also, there is a problem in that the response of the blood circulation of the brain to change in activity is extremely slow, order of magnitude of 1 second. And we know that in 1 second, we can do a lot. So what is going on in our brain? You would assume that when you do a cognitive task, some places are starting to work, and then they interact with others, and then with others, and then with others, and so on relatively fast scale, because in 1 second we can do a lot of processing. So it will be something like you see here. Maybe these places start to interact, and then this, and then that one, and then this one, and then these ones, and so on. If we could reveal such a sequence of interactions or activations which are specific to a given cognitive task, this will be really a new world of looking into what happens in the brain during cognitive functions. The obvious thing is to compare what's going during one function to what's going through another function. Are there common subsequences that mark maybe decision or recalling from memory or things like this? But there are also some theoretical things that would be interesting. Are the sequences involving only cortico-cortical interactions, or are they going from cortex into some deep nuclei, thalamus or basal ganglia, or maybe way to the cerebellum and back to cortex? That's a major question to be asked for understanding mechanism of what's going on. If we take a given process, is it always repeating through the same locations? We have this dogma that if we do something that we think is the same, then the same thing must happen in the brain. For complicated processes, this is not necessarily so. Think of an algorithm running through a network of multiprocessors. So not every time you run the same algorithm, the same processor is doing the same thing. Why not the same in the brain? I can convey, or every one of you can convey the same idea by different phrases, so you reach the end target of conveying the idea via different routes. Does this happen also in the brain when you're speaking about cognitive processes? Maybe yes. And also, if there is a sequence like this, you know that this is my flagship of research. What is the time accuracy of the sequence? Is it very accurate, very sloppy? This may say something about the neural networks behind these processes. So my aim is therefore to expose in front of you a new method that can show the dynamic of activations of cortical areas on a millisecond time scale, and to enable the researcher to differentiate between different cognitive processes. So you can say some sequence is specific for this process and another sequence for the other one. Now, the technique is, I'm sorry, complicated. The goal is fantastic, but the technique is complicated, and you'll have to suffer now for a while through the technique. So first thing, we want to be able to say what are the currents that go through different cortical areas. In EEG, it's voltages. In MEG, you measure magnetic field, but from magnetic field, you can reconstruct currents. Then we want to find some feature in the activity that will mark for us when a cortical process starts to be doing something new. And then, of course, we want to study sequences of such points and then find what sequence is specific to a given behavior. So very briefly, MEG. You all know that, when neurons are active, there are currents flowing through them. And the king here is Idan in this about these processes. In physics, every time there is current, there is a magnetic field around it. If the current goes this way, the magnetic field goes this way. And fortunately, the range of frequency which we are speaking about, which is well below megahertz, then everything is transparent completely. White matter, gray matter, CSF, skull, hairs, and the air around the hair, they are all completely transparent to magnetic fields at this frequency, so we can hope to see things that happen inside by measuring from the outside. Now, for one neuron, this magnetic field is, of course, extremely small, and it goes down very fast, so you will not record the thing. So you cannot really record activity of one neuron outside the brain. But if many of the neurons are synchronized, you can. For instance, 1 million synchronized, like in epileptic fit, make a big spike, which is recordable, and this is one of the clinical usage of the MEG to find from where pre-ictal spikes come in the brain. But even if you have low correlation, say very small, like 0.1, between all the pairs of these 1 million cells, the signal would be only 0.1 smaller, and this is also recordable. So here is how it looks. The subject is lying supine. His head is inside the MEG, which looks like a big hairdresser. This thing has a lot of coils. And as you know, when a magnetic field changes, and a changing magnetic field on a coil induces current in the coil. These currents are replicated here in some little set of transformers called squid, and this is then recorded by the machine. This whole thing, the squids and all the space here and all the coils, are being in a low temperature, like 4 Kelvin, generated by liquid helium. So how does this thing look? So we have many channels. I show you here a few. It looks like something noisy oscillating in some lower frequency, some higher frequency. For the untrained eye, it looks like an EEG. So here are 5 channels. Some of them are neighboring, so you can see that you can see similar wave shapes when they are neighboring. if you look on the power spectrum of this recording, this was recorded at 4,000 times per second. So there are data up to 1,800 hertz, something like this. And you can see this shape. This prominent peak is alpha, it's 10 hertz. Note that this is logarithmic scale. So an octave is the same distance everywhere. The high frequencies is the noise of the machine. How do I know? We can do the same recording without a subject. You can't do it in EEG. But in MEG, you just tell the guy to get out, and you record, and you can see what's the noise of everything. So this is the noise, and you see that above 100 hertz, there's something above noise, and then it rises fast for lower frequencies. To get a recording like this, you need to clean a lot of artifacts, like the main line frequency, all kinds of video cameras inside, motion of the building, the heartbeats, and so on. And we developed, together with Idan, a cleaning algorithm to do all this without using the data itself like other people are doing. And therefore, the rank of the data is not affected by cleaning. So this is already a clean spectrum. OK, now we are interested in what happens in the brain and not what you can record outside the brain. And there are several methods to reconstruct current dipoles inside the brain that can be responsible for the recorded magnetic field. We are using a method called synthetic aperture magnetometry, the software developed by Stephen Robinson. If you are interested in mathematics, get this book of Sakihara about spatial filters for electromagnetic and and EEG brain imaging. It gives all the mathematics for there. And because I cannot understand what's going on there, I will not show you. Now, recently, this guy, Moshe, suggested a method not to reconstruct the currents in one point, but to reconstruct simultaneously the current in several points. And we got this software that does this also from Robinson at NIH. And we came with an idea how to reduce the problem in such reconstructions, which is if 2 points are highly correlated, you may think that the source is in the middle between them. And our idea is something like this. We are interested in points on the cortex. This is the situation. The black asterisks are the position of the coils. We have 248 coils, so like this around the head. The few blue points are shape of the head. You can see here the ridge of the nose. The red points are on the envelope of the cortex. And we put 700 points, roughly equal distances, on the envelope of the brain. We want to know what are the current dipoles in these points. So what we do for every such point, we reconstruct an octahedron around it. And let's see if I can make this work. So you can evaluate more or less what's going on. There is a point in the middle. There is a point in the middle and an octahedron around it. And we solve the equation simultaneously for all the points, 6 on the periphery and 1 in the middle. And we actually relate only to the one in the middle. Why? We think if there are correlations outside this thing, then most of them would be assigned to the activity on the vertices of the octahedron, and the inside will be as far as possible free. So this is a method we developed, and you can find this described well in this paper. How this thing looks, again, something like going up and down. These are few channels. They show currents, the amplitude of the currents in the current dipoles in several points on the envelope of the brain. So this is supposed to be local activity on many brain points recorded simultaneously. Is it true? We take all 248 by 247 pairs of MEG channels. We compute along some 100 seconds or so the correlation between every pair. And this is the distribution of correlations. You can see they go here about 0.8 and here well below minus 0.2. Strong correlations on most of pairs of channels. If we do the same for our reconstructed current dipoles in the cortex, this is the pairwise correlation. So we get something which is much better localized than the external recording. Is what we get really something that we know should be there? So here is an idea by Inbal. She put 10 by 20 by 20, 10 by 20 by 20 targets around Heschel gyrus in the Sylvian fission, primary auditory cortex. She constructed an octahedron around each of them and solved all the equation and got activity in all these 4,000 points, averaged the activity around presentation of short sound beep, and looked for the amplitude of the evoked response. And the dots here is where the amplitude were biggest in amplitude or in significance relative to noise. There are some scattered ones, but there is a clear patch with a radius of 4 millimeters. So let's say this is a centimeter. This is roughly the precision of this method. How does the evoked potential look? This is the time of the start of the beep. You see N100, N100, sorry, and positive wave after it. You see some early responses. This is for the cortical current dipoles. If we take the best MEG, we get something very similar. But if we look on the standard error around it, it's small here and it's much bigger here. So we get a response which is less affected by noise inside the brain, and we localize the place from where this comes from much shorter. It also looks somewhat shorter in time than this thing, because this is affected maybe also by other places in the cortex which are a little bit later. So we know that we can localize correctly responses. They look OK. And therefore, we believe that what we reconstruct is a good reconstruction. Now, what we want to do is study cortico-cortical interaction in some sense. And for that, I'm looking on the following anatomy. Okay? In the cortex, neurons are continuously active all the time. How can we know when they start to process something new? So first of all, cortico-cortical and most thalamocortical connections are excitatory. Most of the synapses in the cortex are excitatory. Most cortical neurons are excitatory. The inhibition is only local, near the dendrites and the neuron and the nerve cell inside the cortex. Therefore, we can assume that when something new comes to the cortex through the white matter, first of all this excitation will build up and activity will rise, and then, because it's a positive feedback, and only later, because there's some delay, inhibition will quench this back and restore activity back, or maybe there would be some oscillations like gamma band oscillations or beta-beta oscillations that people talk about a lot. And example is the thing I've shown you, the evoked cortical response. Something new comes from the thalamus. It makes a big wave, and then a wave in another direction, and then it goes back to spontaneous activity. So we expect that there would be brief activity undulations that mark such instances of when something starts to happen, new starts to happen in the cortex. Now we look on here a small piece of such currents, and you do see some short spikes, not big, a little bit bigger than everything else, but not much. And we assume that these places in time where things like this happen are candidate to place when the cortex starts to do something new. So how can we find them? So we use a method developed long time ago, and most of you who are doing spike sorting in microelectrode are still using this method. We find a template for these things. We decompose the ongoing signal into the projection on the template. and the rest. And the ratio between the two is a signal-to-noise ratio. And we look for places where there's a good projection and also good signal-to-noise ratio. So here is, this is a piece of data from the MEG. We take little spikes or blips like this, or maybe here, and we collect 100 samples. I believe something like this was the masters of Elon for spike terms. So we collect many samples, we align them, we normalize them to be the same amplitude, and then we look for principal components for all these things. There is one needed here covering 96% of the variance. And this is it, this thing up to here. So we can describe well these little blips, many blips, by fit to something like this. We are also interested in things which are not embedded in a sequence of down, up, things like this. Therefore, we add artificially a straight line before the first sample. Mean of everything is 0. Sum of square is 1. It's normalized. And then we look for fits to this thing. Are these things really happening in the brain? So here's what one do. We take a stretch of a recording from one position, 100 seconds of data. There were 340-something events like this. We average them all around the time that we think we detected this blip. The blue trace is this average. Okay? You can see that it's not exactly flat, but it's small periodic frequencies before and maybe after. But then there is a big blip. Each point here is the average of 343 points. So there's also variance around this average. The red line with the scale here is the variance. And you can see, and we could see to our amazement, that during the period of this template, the variance falls precipitously, 2 to 3 times. Down stays more or less down throughout the period of the thing, and then goes up again. Now, what does this mean? It means that what we are used to think of additive noise to our average potentials here is reduced very much. Why? It means that many of the generators, the currents in dendrites, go along with this thing, so the rest of the things there are adding less irrelevant activity, and the variance drop. So this makes us sure that A, the algorithm detects what we want, that B, it really marks that something happens at this moment in the cortex. Now, so you have a machinery, you run it, you get data. Now the times, yes?
Couldn't this be just a result of the fact that this is the event that you're synchronizing on, that you're looking for? So at the time that you're looking for some template, of course, you'll find something which is close to the template.
Right, but if you are setting your threshold such that you'd see about 3 per second, maybe it would not drop that much, right? Okay. So is this related to behavior? So first thing is something very simple. We take an experiment where the subject just lies down and does different things. For 2 minutes is relaxing. For 2 minutes is making mental arithmetics, counting down from 3001 in steps of 7, recalling a movie, things like this. We have 5 behaviors. And for each behavior, we can look on the 700 points, what is the density of these little blips in each of these points. And there is something like this. And it's different looks somewhat different for each behavior, but is it really characteristic? So to test for this, we take the data. It's the number. You see the blue is 200 something, and the red is, I can't read what's there. It's the number of events per 100 seconds. And it's just quantized to 5 levels, because otherwise it would look a complete mess. So we take the 100 seconds of a certain behavior. We divide it into a training set and a test set. So we have 5 behaviors. So from the training test, we have sets. We have 5 maps like this. Then we take a given test set. We compute its map And we look, is it most similar to the map for training set number 1, for training set number 2, number 3, and so on. And if it's most similar to training set number 1, we say it came from set number 1. We repeat this by dividing data at random 100 times, and we get this confusion matrix. Conditional probability that a test set that came from behavior 1, relaxation here, will be assigned to a relaxation. And as you can see, it's mostly on the main diagonal, values close to 1. The sum of all these squares is 4.3, and if this was ideal, it would be 5. This is very good classification. So the rate of this event change as a function of position on the head changes according to the behavior. So they are related in some sense to behavior, these things. Now, we are looking, I remind you at the beginning, for sequences of places that are active, this and then this and then this and then that. And for that, I am going back to something that we did also with a long time ago. Look for repetitive patterns. This is something like a joke we used to tell when we were kids about the teacher to mathematics. He was asked to prove if you have in a room a fire on this end and a kettle with water on this end, prove that you can boil the kettle. So he says, I go to this end, I lift the kettle, I go to that end, I put it on the fire, I wait, and it will boil, QED. Now another question, suppose you have fire at this end and a kettle at this end, prove that you can boil it. So the mathematician says, I go there, I take the kettle, I put it on this corner, and this I already proved that it can be done. So this is something similar. For many years, I was looking for repeating sequences like this in spike trains. Now I'm looking for the same thing in the data from the cortical currents. The experiments in which I'm doing this was supposed to give many repetitions. And this is the experiment now. It's an experiment from the PhD of Idan Tal, in which he trains a subject and asks them to follow the meter of drum beats. So the subject hears something like this: boom, tick, boom, tick, boom, tick, boom, tick, 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 boom. So that changes from 2/4 to 3/4. And he's asked to tap, second finger for the primary beat, third finger for the third beat. So it's going second, third, second, third, second, third, second, third, second. Oops. He hears something else because the meter was changed. Up to here, what he did was sound correctly, but here something is incongruent. Usually, he waits a little and then returns to the new meter. Third, third, second, third, third, second, third, third, second, third, second, third. Oops, again, something wrong. So we now take pieces of data between following a congruent beat, either for a secondary drum beat and third finger, or for the primary ones. We call them congruent pieces or beats. And we take equal number of pieces around the incongruent one, either the secondary beat with the second finger, or the primary beat, or the primary beat with either a secondary beat with a second finger, or a primary beat with a third finger. So we have two sets of behavior, one tapping congruently, and this is the end of sequence, so it's almost automatic. And then something happens, and the person has to realize what goes on and change his motor program. Does this affect the brain activity? You see here one on top of the other, all the 700 channels aligned around the last congruent bit and averaged, and this is how it looks. Behind it in blue, you see the same 700 channels aligned on the incongruent bit. And you can see that there's much bigger activity during half second following the incongruent bit. So definitely something is going on in the cortex always, but definitely something different for the incongruent bit. So now we look for repeating patterns like A, B, C, D, E with equal delays. And we find a lot of them. Some computer memory that we have did not allow us to go below 5. So 5 events repeating twice or more, up to 15 events repeating twice or more within 44 milliseconds. And we hope to see something long that repeats many, many times if we take the same behavior. But we were surprised to see that most of them happen only twice. And there is an issue what to do with that. But something we can do is appreciate the precision. Okay, so this is an idea brought up by Stu Gieman and Eli Bienenstock. Take any event and randomly put it within a window of plus minus W millisecond. If the timing of this event spikes, or in this case, these little blips in the cortical currents, does not matter, then any statistic you would derive from these things would not be affected by the jitter. In spikes, there's a problem of refractory. But for these blips, there's long pause between adjacent ones, so this problem does not arise. So we do this, and here is what we get. If we move them back and forth within 5 samples, this is the distribution of the number of patterns for 100 different random tethering. This is the number that was in the data. If we reduce the tethering to 3 samples, this is the situation. You know that the scale here is different for each graph. If we reduce it by 1 sample, the distribution comes closer to the number found in the experiment, but it never reaches there. Now, the experiment here was sampled at 1,000 hertz, so plus minus 1 sample is 3 milliseconds. So accuracy is definitely better than 3 milliseconds of this repetition. And this is a surprise because we didn't expect in cortex ongoing activity to see things that accurate. There are some patterns that are more specific for behavior. What I want to do is divide again behavior into training and test set and see if there is some matching between the patterns found in the test set to those of the training set. But when everything repeats only twice, you can't do something like this, because either the 2 times would be in the training, so they would not be in the test, or the 2 times would be in the test, so they won't be in the training, or 1 here and 1 here, so you wouldn't know that they happen at all. So we need to find a way to increase the number of repetitions. And I did this in something artificial in the following thing. I said a point is an event like this in a point is the same whether it happens there or in 4 nearest neighbor, because the resolution is not that good. And also, the time, if it's 1 sample before or later, doesn't matter. When we do this thing, we find a lot of things that repeat 3 or more times. And therefore, we can try and divide data between training and test set and see if there is something specific about behavior. And you can see here the little confusion matrix. In 78% test set that came from the congruent bits were classified as congruent bits, and in 87% patterns that came from the incongruent were classified as incongruent. So this is not fantastic, but it's definitely not random. And it shows that there is something in the patterns that has to do with behavior. Now, since we don't see things that repeat many times in one behavior or in the other, we're looking for other ways to see something common. And the idea is that maybe we are losing some of the events because they are small and they are embedded in ongoing activity. So it's a little bit like in molecular biology. You get fragments of something, and you want to reconstruct them into something bigger. So we look for triplets, substructure triplets, within these quintuplets or hextuplets or whatever that repeats many times in many patterns in one behavior and not the other. And as we look on these things, we can see that some of the places repeat many times in these triplets. Like, say, ABC happens, also BXY, also XYB, also ZBU, and so on. So B appears again and again. It's like a hub or a node with high degree in the network. And we want to see where are these hubs. So this shows, I hope, the hubs for the congruent bits. The hubs are painted here in cyan, no, in magenta. And the 4 biggest hubs are all in the cerebellum. Okay, they are showing connections a lot among themselves, other cerebellar points, some with the cortex, and unexpectedly also some with in the pons, which I never thought that the MEG can reveal anything there, but you see it's there. So the hubs for the repetitive congruent things are mostly, or all the 4 highest ones are in the cerebellum. Now, if we do the same for the incongruent bits, you could see that they are not in the cerebellum. 2 hubs are in, hello, where are you? In the contralateral motor cortex, 1 in the frontal lobe, and 1 in the temporal lobe. Completely different places. And this makes sense from the known physiology of routine motion versus recalling a new pattern of motion or something like this. So this supports also the idea that what we are finding is indeed related to the behavior we are measuring. Now we want to reconstruct higher structure, assuming that we are losing some. So suppose we have a quintuplet of events like this, and another quintuplet like this, but they share a triplet. These red things appear both here and here. So we can think maybe these are fractions of something bigger, which has in the middle these 3 things, but on the one side also these 2, on the other side also these 2. So we repeat this thing again and again to find things which come from triplets that were found for the congruent bits and for triplets that were found for the incongruent bits. So we iterate this, and we get something bigger. We never see this whole thing together. We always see some fractions, maybe, and also some things that we didn't see on its own. And we call this something called a super template. So they give really some sequence, this, then that, then this, then that. And the question is the sequence specific to behavior? Because if not, then it's not so interesting. So the answer is yes, but let me show you the sequences. This shows you all the places on the cortex that happen during the congruent bits. Things that happen together are connected by lines, and there are different colors just to ease a little bit the mess. And this is the same thing for the incongruent bits. Now, it's not this many places. The thing is the order at which these things happen. So if we look on a movie with these things, so this is all the things together, but we now show the order at which they appeared. Okay? So first, these 4 or 5 appeared together. Then, after a short delay, these 4 places appeared together. Then, after a short delay, the blue up there. Then these orange ones, and so on. We get a sequence of events, cortical events, that happen together, which, as I showed you, are precise in time, and which we suspect are specific to this behavior. We can do exactly the same thing for the other behavior. This is for the incongruent bits. So this is everything. It looks like a mess, I know. But if you look them one at a time, you see this 4 points, then this 4 points, then the blue one, then the pink one, then the orange ones, the same colors I'm using. Now there was a pause, nothing was happening. Now this, now that, again a pause, the blue ones, and so on. We really see sequences of places that come one after the other, some simultaneously, some alone, some pauses, and so on. This whole thing in real life lasts 80 milliseconds. So this is highly slow motion relative to what happens in humans. Now, the first reaction is, wow, this is complicated. What do we do with something like this? And I'm sorry to inform you that brain processes are complicated. Nothing to do to help about this. But if this were specific, then all the fantastic studies that I talked at the beginning, like comparing different cognitive processes, seeing if they share sub-sequences, where things are happening and who evoked what. Is it cortico-cortical, from cortex to deep nuclei to cortex, and so on. All these things can be now studied. But are these things really specific? So to answer this, I'm going back to the original data. For each such putative sequence of events, I built an analog template. So if this is, let's say, channel number 1 is part of this sequence, I put here the shape of the blip for channel number 1. If after 20 milliseconds channel number 2 happened, I put it here. If then channel number 3 and 4 happen together, I put there. And there are many like this. I'm showing just 4. Now I'm taking the actual data. I'm overlaying this template over the data, and I compute correlation between this shape and the piece of cortical currents in this time, between this and the cortical currents at this time, and so on. I add all together an average and get some correlation here very weak. Now this template is sliding over the data, so I'm getting a correlation that goes up and down. I can take up the highest peaks. What you would see, the 70 highest peaks of this correlation for template that I thought was specific for the congruent, and the position of the 70 highest peaks when the template was specific for the incongruent. And this is how it looks. This is the position of the red line of the incongruent bit. All the black ticks are peaks in the correlation with the template for the incongruent bit. All the red lines are the position of best fit with the template for the incongruent bit. And they are concentrated almost all these before this bit when thing was congruent, and most of all them during the first half second after the incongruent bit. There are very little in the next stage. So the conclusion is, first, the way I massaged the data and what I did is far from being optimal. And there is a lot of room of improving this. But I think I showed it is possible to reveal spatiotemporal sequences of activations that are specific to particular cognitive process. And this opens a whole new door for new studies on what happens in the brain during cognitive processes. Thank you.
What will be the evidence of the ground truth?
Put 700 electrodes over the brain, record directly the activity of the cortex, and compare to this.
Your predictions are also about not entire brain, also
Look, I have 700 points on the surface of the brain.
so to the entire thing. But I'm saying to prove that
few places that they put monkey in a machine that is used for people. And I would not ever permit this to be done. So if I had enough money to have a make for a monkey, this experiment could really be done. It also has to have much smaller bore, because if you go away from the head of the monkey, everything is attenuated along.
Maybe I missed it, but so what about inter-individual variability? Are sequences different for different individuals?
We have analyzed this for several people, but it's still running. So all the details are available for 2 subjects, and the principle is the same, the sequences are different. So that comes to the question, why would you think that when I solve a differential equation and you solve differential equation, the same places in the brain are active in the same way? I'm sure you're not, because I cannot solve differential equations.
Can you show that the variability within a subject is smaller than variability between different subjects?
This will come. I cannot tell you the answer now, but it certainly looks that there's not one of these templates that I try to fish out that do all the fitting. I have to use several templates. And this comes to the question whether what we think the same process, like locking up the new meter of the tapping, is always the same way in the brain, or there are several different routes that can reach the same results. And at this stage I cannot tell you. My personal tendency, because I don't like what everybody says, is that there are many ways to do the same thing, even in the same way.
The same amount of variability you think that exists in the same?
Same, not, but it would be big variability also in one person.
Have you tried such standard repeated tasks of visual recognition, which you know what the person is trying to do?
I did not, but I don't think that for somebody with a good sense of rhythm, to tap like boom, tick, boom, tick, following is much simpler than a recognition task in the visual system, I think, but I didn't. You're welcome to come and do it.
So I just want to reiterate the question about the repetition and variability. I'm confused. I don't know. Maybe the experiment again is undoable because your hypothesis is that I do anything that I do differently each time. So your n is always 1 for the number of repetitions.
You took it too far away. I'm not saying that every time is different, but I'm saying that there are differences. There may be 2, 3, 4 ways of doing the same thing, and sometimes it's going this way, and sometimes it's going that way. And this is a question to be studied. I don't think I've given you here answers how the brain works. I think I've given you a new method that if you use it, or people use it, studying cognitive processes, they can get insight into much deeper and detailed processes than by fMRIs.
Okay. So with this, we close the evening, and we thank all of you for being here. All of you who are here, we thank you for being here, and hope you will come next week, and the next week, and the next week. And thank you, Moshe, for the talk.